Welcome back, everybody. This week we're looking at the Craniata, also known as the vertebrates. Lab 13, focusing this week on the fishes. Craniata, just want to mention that your lab manual uses the term vertebrata. They're essentially interchangeable. They used to exclude the jawless fishes, but now they've been lumped together based on some molecular evidence. So for the sake of this course, they're going to be interchangeable and they mean the same thing. Craniata, vertebrata, same difference. So the craniata are going to be united by several apomorphies, the presence of a neural crest, a cranium, the tripartite brain, more advanced sense organs, a lateral line system, two-chambered heart, liver, kidney, and hemoglobin compounds. Begin with the neural crest. This is a collection of cells that appear near the dorsal margins of a closing neural tube in an embryo. These cells give rise to some of the anatomical structures unique to the vertebrates, including some of the bones and cartilage of the skull. Other things are going to include the cranium, the brain, and the sense organs. So the evolution of an ossified cranium fused with other skeletal components to form a skull. That is kind of self-explanatory. It's going to house a brain and specialized sense organs. The brain is tripartite. Three, partate. It's been partitioned into three parts: forebrain, midbrain, hindbrain. Not the most original names, but hey, it works. So forebrain, midbrain, hindbrain. Sense organs are going to be eyes, olfactory organs, olfactory meaning to smell, auditory organs meaning to hear, and lateral line system, which is super cool. We don't have it, but a lot of the fishes do. So what takeaway from this? Okay, if you're going to have a more advanced brain, it's going to be having all these nice sense organs, specialized sense organs, and more advanced functions. You need to protect it. Okay, how do you protect it? You protect it by putting it into an ossified skull. Okay? It's more delicate, more advanced. You need something that's going to be a little more reinforcing than just squishy body tissue. The rest of the apomorphies, we've got a two-chambered heart that consists of atrium and ventricle. Ancestral hearts did have 4 chambers, but we're looking at the ones that have 2 chambers. Liver is going to act as a chemical factory. Kidneys remove nitrogenous and other waste and maintain the solute concentrations in the body. And hemoglobin is a respiratory pigment present in the blood. Increases oxygen content in the blood. Okay, so what you want to remember is, for the sake of this course, I mean the liver, the kidneys, and hemoglobin, you can spend entire semesters diving into just those functions and what they do. The liver is going to be the chemical factory you want to focus on. Kidneys are going to be removing nitrogenous waste and other compounds, right? Maintaining your dilutions in the body. And hemoglobin, for the sake of this course, is the respiratory pigment in the blood that increases oxygen in the blood. You'll understand why I'm emphasizing that when we talk about myoglobin later on. But again, for the sake of this course, that's what you're going to want to focus on. Some of the members, right? We've got the Minxini, better known as hagfish. They are kind of we used to think that they were a basal lineage. Turns out more recent molecular evidence is showing that they are a degenerate member. So they have lost a lot of the features that we associate with this group. But what you are going to remember about the hagfish: one, they're kind of scary and cool at the same time. So they secrete this defensive proteinaceous threads in the epidermal mucous glands. Right? They've got these keratinized oral plates for grasping and tearing of food. They've got oral tentacles. Right? You kind of see some of that stuff here. Sensory organs for feeding. They are scavengers. Right? That's what those oral tentacles are used for. They have a cartilaginous skull, and they lack the jaws and vertebrae, but they do have a notochord in adulthood. This is why when I say that they're referred to as being degenerate. So we used to think that they never evolved them, but now the more recent evidence of looking at some of the molecular evolution is that they had this stuff there, and way back in ancestors had it, and what we see now have since lost those. I'm going to do a little video in the activity section that looks at these defensive pertinacious threads. They're so cool. Right? They're these super hydrophilic threads. And so picture they can excrete these super thin threads, but as soon as they hit water, they just expand massively in size. And so you can get one of these little hagfish, which is maybe a foot or two long, can secrete liters. I mean, these things are only, 2 inches across at the most, usually closer to being an inch. And they can secrete liters of this mucousy membrane stuff because all they have to do is have a single thread. So picture a piece of like spider silk coming out. It's not spider silk, but something that's thin. But as soon as it hits water, boom, it just expands to hundreds of times its own size. And you'll see that when I do the video in the activity section. But super cool. The rest of the vertebratas, all right, so craniotas as well. Like I said, the term interchangeable based on what we know now. Apomorphes, these neural arches, all right, bones are now made of calcium phosphate, of a photosensory penile organ, which detects light, has some endocrine or hormonal functions, and now we're seeing the presence of myoglobin. Right, so again, if you get into a clinical hematology class, you'll probably spend weeks discussing the difference between myoglobin and hemoglobin and myoglobin. But for the sake of this course, know that myoglobin is the additional respiratory pigment in the blood and muscles for O2 storage. So O2 storage for the muscles, myoglobin, increasing oxygen content in the blood, hemoglobin. And that is a gross oversimplification, but again, for this course, that's what you want to remember. And then we got the gnathostoma and the petrosoma. Yeah, I can't even pronounce that one ever. All right, lampreys and jawed stuff. Moving on. The Petromyzontida, easier to say it's lampreys, Petromyzontida. Okay, so they're going to have this amniotic larval stage, which are going to resemble the cephalochordates that we looked at last week. The adults are eel-like. They have a rasping tongue-like organ and oral disc of keratinized teeth, which you can see here. All right, these are straight out of science fiction horror movies, right? Which they're characteristic of a parasitic lifestyle. What is really cool, right? They have an andromatous life cycle, so adults migrate from the ocean to freshwater streams to reproduce. Very, very, very few organisms on this planet can do that. Most saltwater things have to stay saltwater. Freshwater have to stay freshwater. Certain things like salmon, right? They're pretty famous for being able to go from oceans to the streams. But most things, if you solute concentrations, just would kill them very, very quickly. Just so they can do that, and they're pretty primitive. They lack jaws. Okay, which remember they lack jaws or paired fins. It'd be more obvious when we had a bucket of them that we look at in lab, but they do possess this cartilaginous skeleton. They have lost their bony condition. Right? So again, a little bit degenerate. Apomorphes having this calcium skeleton, they have lost it. Right? But you definitely want to remember they lack jaw, and they do not have paired fins. They got a kind of a one big fin going down the top. They got a bit of one on the bottom, but they don't have the paired fins that you're going to see with the other fish. The anastomatas, all right, so these are going to be your jawed vertebrates. Right, jaws now, looking at jaws with calcified teeth, which evolved from the first pair of pharyngeal bars. Now we're starting to see these paired pectoral and pelvic fins. They provide stabilization and steering while they're swimming. This vertebral column, right, so the vertebra are completely enclosing that nerve cord. So they got an axis of skeleton, provides great strength for the body. It replaces the notochord as the main axis of the body. We're starting to see a little bit more advanced things in terms of digestion. So the stomach, pancreas, the spleen are coming on board. They have heterocercal tails. Okay, what does that mean? You're going to see things with the lobe above the midline of the body is larger than the lower lobe. Think sharks. Right? Sharks are the classic, super clear example of a heterocercal tail. and that is really good for rapid acceleration. This is going to include things like your chondrichthyes and your osteichthyes. Cartilaginous fish, bony fish. Oste, bone, chondri, cartilage. So your chondrichthyes, these are going to be things like your sharks, your rays, and your ratfish. Key things to take away from the chondrichthyes. Placoid, also known as denticulate scales. So it's remnants of the body armor that used to cover some of the ancestral fishes. Right? These denticulate scales, denti meaning teeth, right? If we were to be looking at the shark skin in the lab, which you always would, the scales look like tiny little teeth, right? So denticulate, teeth-like, tooth-like. They look like little teeth, right? So it helps you remember placoid, you get plaque on your teeth. Right? That's the big difference between these and the other types of scales we're going to see on the other fish. So, yeah. They have these pelvic claspers on the males, which is used during copulation, just like the name implies. They're claspers, it's on the pelvis, right? So that's if we were going to be doing the shark dissection, which you need to make sure you watch the video for that. When we would start the dissection, you flip it over, you look at it, and you say, okay, are there claspers or not? If they're claspers, you know you're looking at a male. No claspers, you know you're looking at a female. Possess mostly cartilaginous skeleton, okay? So not a bony skeleton, but a cartilaginous skeleton. And these are going to be things like your holocephalae and elasmobranchi. Don't quote me on how you say those. So the holocephalae, all right, that's going to be the ratfishes. They got those placoid scales, but much, much more sparsely covering their body than you would see on the sharks. They do have these prepelvic claspers on the males, but they also have a cephalic clasper on their heads. Why? We don't know. To the best of my knowledge, they never figured that out. And they have got these tooth plates. Okay, so unlike the sharks, which have these ever-growing teeth that just keep growing, and these individual teeth, they grow, they fall out, it just doesn't matter. You see a shark's jaw, it's terrifying, right? Just this endless rows of teeth. Their teeth have been fused together into a tooth plate, right? They're these weird little fish, kind of weird, kind of cool. But we did have a bucket reserve ones for you guys to look at, but you'll have to look at the videos that I posted into the YouTube playlist. Elasmobranchini, right? Sharks and rays, right? What you want to remember for them, 2 fin spines on their dorsal fins, right? Dorsal, top, dorsal fins. What is definitely the thing to remember about them is they have this ampullae of Lorenzini. It is super cool. Okay, it's this electroreceptor organ located in the nostrils, right? So it's really hard to see even when we were doing the dissections. It's just can't really see it, but it detects the change in the electrical field surrounding the prey. In a very bad analogy, they can smell the electrical current charge being generated by their prey, and not just the field. This isn't just the electricity coming out of their own bodies. Like, your brain is pulsing electricity and your heart fires by electrical pulses. Just when they move through the water, every time, the water flows over parts of the different bodies of these prey, it creates a tiny change in electrical fields, and that they're smelling electricity. Terrible analogy, but it works, okay? They're smelling the electricity around their prey. It helps them zero in on it. Talk about a superpower. How cool is that? We would be doing the shark dissection for you guys if we were here in person. And to answer some people's questions, we would typically get the sharks as bycatch from the docks. These are smaller sharks, typically leopard sharks or dogfish sharks that have been caught by the commercial fishers. They're not large enough to sell commercially, so we would get them and use them for scientific teaching so they didn't just die for absolutely no reason. Right? But make sure that you watch the video shark dissection link on Blackboard, and you have to click to it through the Blackboard link. That way I know you've clicked onto it and you get credit for clicking through there. It's pretty good. It's I want to say it's from one of the public broadcast universities in Washington, but make sure you watch that. It's very informative. osteichthyes. The osteichthyes, or the bony fish. Right? Remember, I've been telling you guys all semester to learn what these root words mean. Okay? Oste, bony, ichthyes, fish. So ichthyology, study of fish. Oste, bones. Right? Osteoporosis is bone disease. You'll learn when we look at manatees in a couple weeks. Pachyostonic is how you define their bones. Right? Pachythick bones. They got thick bones. Right? Moving on. Osteichthyes, the bony fish. They have a gas bladder or gas lung, depending on when you're looking at, that helps them regulate buoyancy. Right? So it's just this air bubble inside of their not chest cavity, but their gut cavity that helps them maintain zero buoyancy, so they can be up or down in the water column wherever they want to be. Their scales are adenticulate. Yeah, the sharks had denticulate scales. These are adenticulate, meaning when you look at them, they look nothing like little teeth. They possess bony skeletons, all right, no longer this cartilaginous skeleton. They got the operculum, right? The gills are covered by a hard flap, right? Gills, very sensitive, need to protect them, so they got this big bony flap, right? So draws water across the gills, but also protects them. Sharks, open gills. I am gesturing with my hand, which you guys obviously can't see. All right, so that's going to be things like Actinopterygii and the Sarcopterygii. So the Actinopterygii, ray fin fishes. Ray meaning, if you think of like the sun rays you used to draw as a kid, you got the little sun in the corner. Anyway, if you look at their fins, right, coming out like rays. Right? And their scales are ganoid scales, meaning that they're composed of ganoin, right, as opposed to the shark scales. So they got these adenticulate ganoid scales. Right? Ganoin is just an enamel-like tissue in the scales, earliest evolving these things. Right? Most have a homocircle tail as opposed to the heterocircle tail, meaning that you take the midline of the body, tail, same size top, same size bottom. So the caudal fin is symmetrical. Caudal fin. That is a caudal fin. Symmetrical, same top, same bottom. Right, and these are those ganoid scales. Right? They do not look like little teeth. And the Sarcopterygii, right? Muscular lobed fins, right? This is a coelacanth. They have crazy true enamel on our teeth, so kind of like us. Coelacanths, how do I explain this? We didn't think they existed for a very long time outside of the fossil record until early 1900s. I want to say it was the 30s. Somebody was in a fish market, saw one laying there for sale as just some other type of fish. They happened to recognize it, and it's like, this should not be a living thing because this thing went extinct with the dinosaurs, and that would be like going to some random meat market, and you're like, oh, there's a chicken, that's a velociraptor, that should not be alive, and yet, here it is. Turns out they were just really good at hiding from us, at least from the scientific community, and so they're still alive and well. So their gas bladder has been ossified, so they still have it, but it's completely non-functional. And they make these things called biosalts, which are produced by the liver, which is part waste, part digestive juice. All right, and this is going to include groups things like your dipnoids, all right, so those are your lungfishes, and tetrapods. Tetrapod meaning 4 feet, and that goes back to these muscular lobed fins, right? What is a tetrapod? We're tetrapods. Okay, the lungfish, again, during the activity section, I'm going to be showing you a nice, cool video for these things. But they use their gas bladder for air breathing, not buoyancy, and they have very thin scales on their skin. They get their name lungfish because they can air breathe. And really cool video I'll show you guys is that they live in places that can periodically completely dry out. So if you're in an African lake somewhere, swimming around, everything's good. Dry season comes, your lake disappears. What do? They bury themselves in the mud, make a mucus cocoon, right, which turns this leathery substance, and they basically just wait and hide, and they start breathing air. And they can last for years doing that if they need to, and they just slowly start to digest their own body. And then as soon as enough water comes by, they will rehydrate themselves and then slither on back and get back to what they were doing. So that's pretty cool. That's the end of the lecture. Okay, so make sure that you watch the activities video and click through the links that are in the Lab 13 playlist. You need to use the link for the shark dissection. It's an unlisted video, so if you want to get credit for watching it, you got to click through the Blackboard link. Practical 5 is this week. It'll be following the same format as before, meaning it's open for 24 hours. You have 30 minutes to complete the exam once you start. You get one shot at it. No open book, no open notes. Can't really enforce that, but it's just kind of honor system on that one. But you've only got a very limited amount of time, so if you're trying to look up every answer, you will miss something. The last 2 questions I will be manually grading because they're short answers, so don't expect a grade right away. It'll take me a couple days to grade all of them, and I will get to that. And that is the end of the lecture. Good luck. Miss you guys. Stay safe and take care.